All right. So microbiology. So chapter 16, innate immunity. Okay. So sure you guys are wondering after all these chapters, like why aren't we like dying of everything that's infecting us? And the reason is immunity. Okay. Um, Immunity is broken down into two components: the innate immunity, which is the first two barriers, and then the specific immunity, which is the not which is the third barrier, which is the one that's most talked about. Okay. All right. So I'm sure you've seen this. This is a white blood cell right here. That's phagocytosing a spore. Okay. Uh, okay. So this is a really kind of important big picture. Okay. So viruses, fungi, bacteria are on the outside. Okay. Outside of the body, it's also the inside the stomach and stuff like that. Okay. All of that is the outside of the body. Okay. The first line of defense is the skin, the mucous membranes, the antimicrobial substances that are spread on our skin, or mucous membranes, um, the IgA antibodies, etc., etc. Okay, if they ever get past this first line, they encounter the second line of defense, which is inflammation, fever, phagocytes. Okay, all these are again non-specific, and then finally we hit the third wing, which is the humoral and cellular immunity. Okay, so here are the cells that are involved. Okay. Okay, white blood cell count. Okay, is a measure of leukocytes. Okay, um, high white blood cell counts um, is either a bacterial infection, an autoimmune disease, or side effect of medications. Also, high white blood cell counts can indicate leukemia. Okay, low white blood cells can indicate viral infections, pneumonia, downregulation of the white blood cells, which is another type of autoimmune disease, and or cancers. Okay, so that's the white blood cell count, about 5,000 to 10,000 white blood cells per cubic millimeter. Okay. And out of those, this is the normal range. Okay. Okay. So, number one, 16.1 is kind of important. So, um, okay. So definitions, right? So these are definitions. Okay. So innate immunity, you're born with it. That's innate. Adaptive immunity, you have to adapt. Adaptation takes time, so adaptation will always be slower to respond, but the payback of it is that it has a memory component. Okay. Okay, TLRs, toll-like receptors, are host cells attached to pathogen-associated molecular patterns. Okay, and that induces the release of um, some hormones called cytokines from the host cell that regulate intensity and duration of immune responses. So, nope, or actually, uh,
Host defenses consist of two categories, the innate or non-specific defenses and the adaptive or specific defenses. The human body has many innate defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system. The human body has two main divisions of adaptive defense, humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses.
All right, so there you go. Make sure you can answer those questions. Okay, so the first layer of defense is skin, okay, and mucous membranes. Okay, so make sure you understand all these three learning objectives. Okay, the dermis, epidermis. Okay, so skin, okay, is made out of the epidermis on the outside, and then the dermis on the inside. The shedding and dryness of skin actually prevents microbial growth. So microbes grow on it, but we shed it off. And the dryness factor and the acidic factor actually helps the skin as well. Okay, most microbes can't grow in this dry environment, dry acidic environment. Okay, this is off the palm of your hand or something like that, so you can see like there's huge layers of dead tissue, and viruses can't infect that, cells, bacteria can't get through that, etc., etc., etc. Okay. And the keratin. Okay, the keratin makes it waterproof. So again, making it waterproof prevents water from going through, which prevents a lot of bacteria from going through. Okay, all right. Mucous membranes. Okay, the mucous membranes prevent the tract from drying out, but the mucus is also sticky, and therefore they trap the microbes, and they usually take it to the GI tract, which you swallow and you don't worry about. Okay. Okay. The lacrimal apparatus. Okay, so your eyes can actually get infected relatively easily without the lacrimal apparatus. The lacrimal apparatus. Okay, so you are here. Okay, these are lacrimal glands. What they secrete is fluid, and then when you blink, your blinking actually wipes the fluid across the eye, so you wash out all the bacteria. In addition to that, there's lysozymes in there that actually break down bacteria, and then you wash it into the nasal lacrimal ducts, which connect to your nose and eventually out the body. Okay. All right. Other physical factors: the ciliary escalator. We talked about this anatomy as well. It transports microbes trapped in the mucus away from the lungs and into your throat. Okay, earwax. The reason why you have earwax is pretty straightforward. Okay, they prevent microbes from entering the ear. Okay, urine cleans the urethra. Okay, vaginal secretions move things out of the vagina, etc., etc. So this is a ciliary escalator. Okay. Okay. The chemical factor sebum forms a protective film. Okay, and lowers the pH of the skin. This is the oil, right? Lysozymes are in perspiration, sweat, tears, saliva, and destroys the bacterial cell walls. The low pH, okay, of the stomach destroys both bacteria and toxins. The lower pH of the vagina also inhibit microbes. That pH does, the pH secretion of the vagina does change depending on conditions. Like if you're ovulating, it actually increases the pH. So that's where you can actually get infections. Okay. Okay. Normal biota also competes with pathogens via microbial antagonism. Okay. Okay. That is an important thing. Okay. Competitive exclusion. So the normal microbiota are located already in spots where other bacteria want to come in, but since they are already there in the first place, they have squatting rights basically, and they kind of already there. So for other bacteria to get in, they have to push these guys out of the way or kill them or some other format of getting them out of the way, which is difficult. Okay, these normal biota also produce substances that are harmful to pathogens. Okay, like they lower the pH, etc., etc. Okay, so commensalism, one organism benefits while the other is unharmed. That's what a lot of the normal microbiota is for us. Okay, they benefit from living on us. Okay, while us, the host, is unharmed. Okay. Okay, probiotics are live microbial cultures. You've probably seen them associated with yogurts and stuff. Okay, that are administered to produce a beneficial effect. Okay, so these are like germ-free mice. All right. All right, the second line of defense. Okay. Okay. Classified leukocytes describe the roles of granulocytes and myelocytes. Okay. Describe the 8 different types of white blood cells and name a function for each type. Okay. Okay. So, form elements in blood, red blood cells, white blood cells, and platelets. Okay. So, white blood cells are leukocytes. Leukocytes split into two groups. Sorry. They either have granules inside them or no granules. So, granulocyte have granules, agranulocytes, no granules. Okay, and they break down into three groups. Neutrophils have granules that absorb both the acidic dye and the basic dye. Okay, and so they appear purple. Okay, they're phagocytic. They work in the early stages of infection. Okay, they are phagocytes, which means they eat up bacteria. Okay, basophils will appear red because they absorb the basic dye. Okay, they release histamine. They work in allergic responses. Okay, the last one is xenophils. Xenophils like the eosin, which is the acidic dye, and so they will appear reddish. Okay, basophils are purplish, the eosinophils are reddish, and what they do is they're phagocytic as well, but they're toxic against parasites and helminths. Okay, what they do is something slightly different than other ones. Okay, they basically deal with groups of pathogens that are too big for the other blood cells to deal with. Okay, so phagocytes will engulf other cells. To engulf another cell, you have to be bigger than it. Worms, okay, are about 1,000 cells big, so they're much bigger than neutrophil or macrophage. Okay, so what these guys do is they kind of attach themselves to these parasites and they release chemicals. That's what the granules are for, that blow holes in the worm and the worm dies. Okay. Okay, the agranulocytes, okay, are leukocytes with granules in their cytoplasm. They're not visible with a light microscope. Okay, which means the granules are too small to see. Okay, monocytes mature into macrophages. Okay, where they become phagocytic. Okay, um, okay. Dendritic cells are found in the skin, mucous membranes, and thymus. Okay, they are phagocytic. These guys are made in the blood, but they come out really early in development, and they're found in the skin, in the epidermis. And what they do is they kind of sit around and eat things, and then present what they eat to these other cells, the T cells, B cells, and T cells and B cells. Okay, and the last group is the lymphocytes, T cells, B cells, and natural killer cells, NK cells. These guys play a role in adaptive immunity. Okay. Okay, the lymphatic system. Okay, so this is your lymphatic system. Okay, this is your lymphatic system. Basically, these are one-way valves where fluid drains in, and once they drain in, okay, they get carried throughout the body. Especially important are the lymph nodes and stuff. Make sure you can.
Host defenses can be divided into two categories: innate or non-specific defenses, and adaptive or specific defenses. These defenses act together to protect the host against infection. The human body has many innate or non-specific defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, plus the second-line defenses such as phagocytosis, inflammation, and the complement system. All of these defenses act against all pathogens and are not specific to a given pathogen. The third line of defense is comprised of the adaptive or specific defenses. These defenses recognize a specific pathogen and must be activated by antigen-presenting cells before they can work. These defenses include antibodies and cytotoxic cells. Together, these defenses make it very difficult for pathogens to penetrate, colonize, and cause disease. Host.
All right, so make sure you know what these are. Okay, so the lymph nodes. I don't think they show you a picture, but these are the lymph nodes. The lymph nodes are where the B cells and T cells actually fight off pathogens. Okay. All right. Okay, phagocytes, cellular eating. Okay, fixed macrophages are residents in tissues. They're usually named after whatever tissue they're in. The ones in the lungs are called dust cells. The ones in the liver are called Kupffer cells, and other cells. Okay, and they eat bacteria. So you can see that happening here. Okay. Okay, the mechanism of phagocytosis. Okay, chemotaxis, chemochemistry, taxis, movement. So chemical signals. Okay, so what phagocytes do is something called positive chemotaxis. They move toward certain chemical signals, and that usually leads them to the microorganism. Okay, adherence is attachment of the phagocyte to the surface, and an ingestion is the swallowing of the microorganism. Usually that's helped by something called opsonization, okay, in which the microorganism is coated with protein antibodies or complement other factors that attract these phagocytes, and that makes the swallowing and adherence easier, and that makes it easier to ingest. And in digestion, is the microorganism is digested inside a phagolysosome. So what happens is the ingestion is the wrapping around of the organism, and it becomes put inside a phagosome. To the phagosome, you attach a lysosome and become phagolysosome. The lysosome will have a lot of chemicals that are designed to break down the bacteria. Okay, so here, okay, adherence, okay, ingestion, okay, the phagosome, the lysosome attached to the phagosome, which then causes the digestion of the components. Okay. And then um, after you break it down, you actually release all this waste material. And the other cool thing you do, which they don't tell you about, is they display some of it on their surface. So on the surface of all these white blood cells are like the trophies of everything that's ever killed, which is pretty awesome thing about. Okay, so that's this. Okay, we'll watch this video and then we'll cut this off and have part two because this chapter is pretty long.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some protozoans for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils and are collectively called phagocytes. Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation.
Okay.
Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
More videos.
The human body has many surveillance mechanisms for detecting pathogens. Phagocytes are able to detect foreign cells by looking for molecules on the surface of those invaders that are not found in the human body. Adaptive defenses, such as B cells, antibodies, and cytotoxic T cells, are looking for a specific antigen on the surface of the invaders. If a microbe can hide from these surveillance methods, then it's more likely to reproduce within the body. When immune surveillance cells, such as macrophages, are looking for invaders, they search for surface molecules that are not found on host surfaces, such as peptidoglycan. Capsules that surround some bacteria are usually composed of polysaccharides similar to molecules found on host cells. Thus, when a macrophage encounters an encapsulated bacterium, it doesn't recognize it as foreign and ignores it. This is a strategy used by Streptococcus pneumoniae, a common cause of bacterial pneumonia. Even if a bacterium is engulfed by a phagocyte, it may not necessarily be killed. The tuberculosis bacterium is able to survive within a phagocyte by preventing the fusion of lysosome and phagosome. This keeps the digestive enzymes away from the bacterium. Bacteria such as Shigella and Listeria can actually escape the phagosome and live within the cytoplasm of the phagocyte. Even if the phagosome does fuse with the lysosome, some microbes are not affected by lysosomal activity. Leishmania, a protozoan, is able to resist the lysosomal enzymes. Legionella and some staphylococci inhibit the pathway that leads to oxidative killing within a phagocyte. Some bacteria, including the tuberculosis bacterium, actually reproduce within the phagocyte. Several different types of microorganisms are capable of hiding from host defenses by changing their surface antigens frequently. These changes may occur as a result of mutations or genetic recombination. RNA viruses such as influenza virus or HIV frequently exhibit antigenic variation on their surfaces. Trypanosoma, the organism that causes sleeping sickness, has a surface glycoprotein that undergoes frequent changes. In all of these cases, it is the survival of the fittest. The microbes with the undetectable antigens are the ones that are least likely to be detected and most capable of reproduction.
Okay.
Successful pathogens have evolved methods for inactivating the host defenses that would normally lead to the pathogen's destruction. This gives the pathogen a better chance of colonizing and reproducing within the host. Complement is the group of proteins that, when activated, undergoes a cascade of reactions leading to inflammation, opsonization, and cell lysis. Some bacteria simply evade complement by covering the complement binding sites with a capsule. Other bacteria have peptidases that destroy key complement proteins and block the cascade. Neisseria and some viruses mimic a control molecule that the human body uses to keep complement inactive when it's not needed. Many bacterial pathogens, including Pseudomonas and Group A Streptococci, produce molecules called leukocytins. Leukocytins cause destruction of phagocytes, such as neutrophils and macrophages. Species of Neisseria that cause meningitis and gonorrhea secrete an enzyme called IgA protease. This enzyme destroys a class of antibodies called IgA that are located in mucous membranes. As a result, these bacteria are much less likely to be detected in these regions. Another way in which a pathogen can inactivate host defenses is more general. Several viruses, most notably HIV and the measles virus, are able to cause generalized suppression of the immune system. Often, these viruses work by suppressing the activity of cytokines or by reproducing within macrophages and T cells. Some bacteria produce toxins that are superantigens. These toxins distract the adaptive defenses, causing them to produce a non-specific exaggerated response and fail to respond to the actual invader.
Cool. All right. So make sure you guys understood all that. Okay. And then I will see you guys for part 2.